Well, good morning and welcome to the third lecture, the second half of Genome 361. So today we're going to continue with the theme of linkage and making genetic maps, but we're going to talk about a different kind of marker that we're going to use to make our genetic map. So most conventional mapping exercises involve looking at polymorphic DNA sequences. These are sequences that vary in the population, and we use those to map genes. We map genes relative to polymorphic sites where there's variation again in the population. And the primary method that's used for mapping is, it just involves hybridization, which I'll define in more detail when I get into the lecture. So the photograph at the bottom, on the bottom left, shows a picture of Nancy Wexler, who played a critical role in helping to map the gene that's involved in Huntington's disease, which is a really severe neurodegenerative disorder that affects people that typically are in their 40s. And she basically identified a large pedigree in South America and a large group of families, related families. And she used the information in that pedigree and used molecular markers to map the location of the gene. And once you map the location of a gene, you can ultimately use that information to identify the gene. I'll kind of illustrate that principle on the next slide, and then we'll go through the techniques that are involved in mapping or genotyping molecular markers, I should say, and then how you can use molecular markers to map genes. We'll go through some of that logic as well. Okay, so in the last lecture, I told you one reason why genetic maps are useful, and the reason I told you, the reason I gave you in the last lecture is that it helps you to predict the outcomes of crosses. It helps you to figure out roughly what progeny you're going to get and in roughly what proportions. Another reason that genetic maps are extremely useful is that it helps us to identify genes that are involved in a heritable human disease. So for example, I'm showing you here a pedigree that is segregating a heritable form of Alzheimer's disease, which I think probably everybody's familiar with. It's just a severe neurodegenerative disorder that affects typically the elderly. But there's also genetic forms. In most cases, we don't really know what causes Alzheimer's disease, but there are some rare Mendelian forms of Alzheimer's disease that are caused by gene defects in specific genes. So if you look at this pedigree, based on information you learned in the first half of the class, it basically says that there is a mutation in a single gene that seems to cause this gene. And you can also look at the pedigree to infer a little bit about the inheritance pattern. So for example, the inheritance pattern seems to be consistent with an autosomal dominant pattern of inheritance. So again, I'm just telling you this, but you should be able to look at the pedigree and infer that for yourself based on what you learned in the first half of the class. Okay, so at this point, we know that there's a mutation in a specific gene somewhere on one of the autosomes. So, but the question is, where is this gene that is involved in causing this Alzheimer's disease trait? Where is it located, and how can we identify where the gene is? So, as people all know, there are 22 autosomes, and there's an X and a Y chromosome, the sex chromosomes. And based on the pedigree, we can only rule out the X and the Y chromosomes because it doesn't show a sex-linked inheritance pattern. So the gene could actually reside anywhere on any one of these autosomes. So let's just imagine that the gene happens to reside on chromosome 9, and let's say it's at the very tip of chromosome 9. So the question you're asking when you try and map a gene is you're basically asking, do markers of a particular allele of some gene, do they seem to show a segregation pattern that's consistent with linkage to the gene that you're interested in trying to figure out where it is? So, and I can just ask you a few hypothetical questions. Typically, I would do this in a classroom full of people, so I would just let some people try and answer the question for me. So, but I'll just go through this anyway. So the question is, with markers that reside here exhibit linkage to the Alzheimer's disease trait, and by markers I mean, would alleles of a particular gene that happens to map on chromosome 6, would those show linkage to the Alzheimer's disease trait? And hopefully, based on what I told you in the past two lectures, your answer would be no, because genes that are on separate chromosomes assort independently. This is basically what you learned in the first half of the course. So let's ask a different question then. What if you had markers that mapped to the opposite end of chromosome 9, opposite end from where the Alzheimer's disease gene resides? Would those exhibit linkage to the Alzheimer's disease trait? And I just wanted to let you know that the chromosome 9 consists of 140 million base pairs of DNA. So the marker would be, let's say, 120 million base pairs away from the Alzheimer's disease gene. So the question, would markers there exhibit linkage to the Alzheimer's disease trait? And once again, hopefully your answer is no. And it's because what I told you last time is that genes that are more than 50 centimorgans apart are just going to behave as if they're sorting independently, just as if they were on a different chromosome. So only genes that really reside close by to the trait of interest are going to exhibit a pattern of inheritance that's consistent with linkage. So finally, I'll ask, what about markers that are actually right next door to the Alzheimer's disease related gene? Would those exhibit linkage to the Alzheimer's disease trait? And in this case, hopefully your answer is yes. So basically, the strategy people use to identify roughly what chromosome a disease gene resides on and sort of pinpoint what part of the chromosome that disease gene resides in is that they just identify linkers that they identify markers that are linked to the gene of interest, and then that gives you basically the information you're after. It tells you that the gene is on that chromosome, and it tells you roughly where that gene resides on that chromosome. Okay? So just a little bit about genetic markers. I've basically been talking about genetic markers for a little bit. They're just basically inherited variations that are used to test genetic hypotheses. So for example, the wing and eye phenotypes we discussed previously in our examples in lectures 1 and 2. Basically, the only difference between the purple plus and purple mutant allele is that the mutant allele has some alteration in the DNA sequence that gives rise to the mutant phenotype. So we're actually, in the crosses I've talked about in the past, we're just looking at the visible traits that you can observe by looking at the flies to say whether you've got the purple wild type allele or if you're homozygous for the purple recessive allele. But you could also look at the DNA sequence itself to say what the genotype of these flies are. And the same logic holds for the vestigial gene. In all the past examples we talked about, we just looked at the visible traits that are conferred by these alleles. But you could also look at the DNA and distinguish these two genes just from looking at the DNA sequence, because the DNA sequence is not going to be identical. Okay, so we could also apply this same process to humans by just looking at some visible traits that are known to be basically simple Mendelian traits in humans. So for example, hanging versus attached earlobes, that's a fairly simple Mendelian trait. Also, a widow's peak is also a simple Mendelian trait, and there are many other simple Mendelian traits in humans. But the problem with this in humans is that it poses some serious challenges. And one of the challenges with this is that there are a number of these traits, but they're actually relatively limited. You're not going to find these visible traits that have simple Mendelian inheritance patterns. The genes for them are not scattered all over the genome. There's a fairly limited set of them. And then also, because you don't define the crosses that you're setting up, you just have to live with what you see, and oftentimes people don't have enough of these traits to really be effective as mapping tools. So we have to develop some better markers for mapping. So on the next slide, I'm going to summarize many of the challenges that are associated with mapping genes in humans, but I'm just going to focus on two of the challenges today in today's lecture. So this is, again, a list of all the challenges you have to deal with when you're trying to map genes in humans. So it turns out many traits are polygenic, which means that the traits have multiple genes that contribute to that trait. Another challenge is that there's variable penetrance and expressivity. These are terms you talked about in the last lecture. So somebody might have the genotype, but they either don't exhibit the phenotype, or the phenotype varies considerably depending on the genetic background of that individual. It's also much harder to know the parental type. For example, you can't do a test cross with a human, right? So it can be much more difficult to know what the parental type is in humans. Another challenge with humans is, unlike the examples I talked about with flies, where we're looking at 400, 500, 1,000, 2,000 offspring, the pedigrees in humans are quite small, so your level of confidence is much lower. So there are statistical methods people have developed to deal with that part of the genetic linkage studies in humans. And in the past, I used to talk about these statistical approaches, but this is not used very often anymore. So this year, I've decided I'm not going to talk about that, and instead I'll spend time on something that would be more profitable to you, that's more relevant. Okay, so finally, one of the first of the two things that I plan to talk about today is there just simply aren't enough markers. Basically what I told you on the previous slide, there just aren't enough markers that are easily scored, that produce easily scored phenotypes, such as hanging versus attached earlobes, or the ability to roll your tongue, or hair color. In fact, hair color is not a very simple genetic, a Mendelian genetic trait. So there just aren't enough really simple Mendelian traits that you can that are present in humans to be useful in mapping genes. And then another challenge with humans is many meiosis aren't informative. I'll define that when I get to that point later in the lecture. So again, the two things I plan to talk about in today's lecture are the last two, number 5 and number 6. There aren't enough markers to produce easily scored phenotypes, so we have to deal with that problem. I'll talk about the approach people have developed to deal with that problem. And then at the very end of the lecture, I'll talk about what a informative versus non-informative meiosis is. Okay, so the solution to the marker problem is that people have been using in humans, and in fact this is the standard approach in model systems as well, is that they develop molecular markers that they use to see if if a particular site on the DNA is linked to the gene that you're interested in trying to map. So again, instead of using observable traits, perhaps you can just look at differences in the sequence of DNA, places where you know there's variation in the DNA sequence in different individuals in the population, that you can just basically detect with established methodology that's currently in use. These polymorphic molecular markers are the primary type of marker that's used in contemporary genetic studies, again, both in humans and in model systems. So the basic idea here is that let's say that you have a stretch of chromosome we're looking at, and let's say that these yellow boxes represent genes along the length of the chromosome. Basically, if you compare the sequence from any two individuals of this portion of the genome, what you'd find is that there's a number of sites where the sequence would vary from one individual to the next. And in fact, on average, when you make this kind of comparison, what you see is typically there's a site that varies in the genome 1 every 1,000 base pairs between two different homologous chromosomes. So this variation is spread all over the place. It's very closely spaced, so there's lots and lots of it available to use in mapping genes. It's very easy to test for these things. We'll spend a lot of time in today's lecture talking about the methods that are used for testing for these things. And it's not limited to just genes. So this variation exists within coding sequences, but it also exists everywhere. It exists in the regions that are in between coding sequences. It exists in introns. It exists in promoters. So the variation is all over the genome. So it can basically be used to map genes pretty much anywhere in the genome. So let's just define what a polymorphic marker is. So a polymorphic site or locus is just simply a location in the genome where at least two different versions of the sequence exist in the population, each at a frequency of at least 1%. And the reason why we put this threshold of 1% on here is because if the frequency of the variation is much less than 1%, it actually doesn't end up being very useful in mapping, because in most cases, when you look at that site, most people are going to have the same sequence there. So you need to have variation at these sites they're using as markers for mapping to really detect whether there's linkage to the trait of interest. Okay, so let's consider the UW student population. There's about 40,000 UW students. So for example, amongst this 40,000 students, that would mean that there's about 80,000 copies of chromosome 2. Okay, so each person in the population has 2 copies of chromosome 2. So that means there's 80,000 copies of chromosome 2 in that 40,000 student population. So let's just say at one particular site, if you mapped if you sequenced a portion of chromosome 2 in all these 40,000 students, maybe you would find that 70,000 copies of chromosome 2 have an AT base pair at this site, and 10,000 copies have a CG base pair at this site. So that would represent a polymorphic site, because each is present at a frequency of greater than 1% in the population. And again, it varies in the population. There's a different sequence there in different homologous chromosomes at that particular site. So there's lots of different types of polymorphic molecular markers that can be used in these mapping experiments. So the simplest type is single nucleotide polymorphisms, oftentimes abbreviated by this acronym SNP. And basically these are just sites where if you compare two sequences in the genome, it's just like the one I showed you in the previous slide, where there might be a base pair change that distinguishes the two homologous chromosomes. So in this case, some members of the population have a T at this site that's highlighted, and some members of the population have a C at that site. Another type of polymorphic molecular marker is insertions and deletions. So in this case, I've highlighted in red in some individual populations somebody has 2 consecutive Ts, and in other members of the population, the members of the population have 3 consecutive Ts. So depending on how you look at that, the red T above that particular allele could represent a deletion of 1 T, or the allele below on the bottom could represent an insertion of an extra T. But the point here is that there's variation in the number of nucleotides at a particular site. These are again known as insertions or deletions, or sometimes they're abbreviated indels. And then a third type of polymorphic molecular marker that's used in mapping genes are called variable number tandem repeats, oftentimes abbreviated VNTRs. These can actually have other names as well. So another name that's often used in the literature is microsatellites. So for example, I'm showing you 3 different alleles here. And basically, each of these alleles has a repeat of a CAG sequence. And you can see that's repeated again and again. And basically, if we designate these alleles by the number of repeats they have, we would say basically the first allele has 5 repeats, the second allele has 6 repeats, and the third allele has 7 repeats. So it turns out there are many of these VNTRs in the human population, and they're highly polymorphic. They can differ greatly from one individual to the next in the number of repeats that are present. So these are the 3 main types of polymorphic molecular markers that are used in mapping genes. And so I'm going to spend most of the rest of the lecture talking about the methods that are used to detect these polymorphic markers. So how are they detected? So the genotype methods for detecting polymorphic markers, typically there are 3 main approaches. One is polymerase chain reaction, another is hybridization, and the third is sequencing. So actually it turns out hybridization is a feature of all 3 of these methods, but I'm listing hybridization separately from the other 2 because it involves purely hybridization. The other methods basically just involve an element that involves hybridization, but there's other parts of the process as well. So I'm only going to talk about 2 of these things, the polymerase chain reaction and hybridization today, and we'll talk about sequencing in a later lecture. So the polymerase chain reaction is really only useful for identifying indels and VNTRs, and I'll demonstrate that in a subsequent slide. And hybridization and sequencing, these are the main approaches that are used nowadays for mapping these genes. And mostly these are the primary methods because they're very easily multiplexed. In other words, you can look at many polymorphic sites simultaneously using these methods, whereas the polymerase chain reaction is typically only useful for looking at one polymorphic site at a time. So I'll start off by talking about the polymerase chain reaction. When I'm finished with that portion, I'll go on to the hybridization methods that are used for mapping polymorphic sites. And then again, as I mentioned, I'll talk about sequencing in a subsequent lecture. So I suspect probably a lot of you already have heard something about the polymerase chain reaction, either from some other course you've taken, or if you've worked in a lab, you've probably used it because it's commonly used for lots of different purposes in molecular biology. So just a little bit about the elements of the polymerase chain reaction. So it uses a DNA polymerase. It can make unlimited quantities of the DNA of interest. It only requires a single template molecule, so it's very sensitive. You can start from a single molecule of a DNA sequence and produce, many copies of that molecule very quickly. And it uses 2 DNA primers. Again, I'll define all of these things in the subsequent slides. Okay, so a little bit about the reagents that are typically used in PCR. So you have the target DNA. Oftentimes it's genomic DNA, but you can use lots of different sources of the DNA. And you can have as little as 1 copy of the genomic DNA. And then you also need 2 specific primers, typically termed left and right primer. I'll have more to say about that in just a little bit. And these primers are short oligonucleotides that are complementary to sequences on either side of the region that you want to amplify using this polymerase chain reaction. So the term oligonucleotide refers to just a few. And although in my examples, I'm going to be showing you primers that consist of really very small number of bases, in this example here, both of the primers are 4 bases in length. In fact, in a real PCR reaction, you'd want your primers to be 17 to 20 nucleotides in length. I'm using really short ones in my examples just to save myself from having to write out the entire sequence. Another thing that you want to have in your PCR reaction is all 4 DNTPs, ATP, CTP, GTP, TTP. These are the reagents that are used for synthesizing a new strand of DNA. And then to synthesize a new strand of DNA, you need to have a polymerase. And the one that's typically used in the PCR reactions is a DNA polymerase from the heat-loving bacteria, Thermus aquaticus. And the reason you use this polymerase from this particular organism is because in the PCR reactions, as you'll see in the subsequent slide, that you will actually have to go through repeated processes of heating and cooling, and most polymerases would basically be denatured by the high temperatures that are required for doing PCR. But the polymerase from Thermus aquaticus is quite stable, and so it survives under very high conditions or high temperature conditions. Okay, so let's just talk about what's involved in doing PCR. So let's say this is your starting double-strand DNA molecule. The first step you want to do is you want to heat it up, and what that does is it causes the DNA to denature, and what that means precisely is the two strands separate from one another. And then what you do is you anneal or hybridize your primers onto the DNA. So basically you have primers that are complementary to regions of interest, and they span the region that you want to amplify. And then you cool the DNA down with these primers present, and they will hybridize or base pair to the corresponding sequences in the DNA. So here's your two primers. They've actually hybridized to the portions of the DNA where you want them to hybridize to. And then what you do is you synthesize DNA, basically. You have all 4 DNTPs, you have the polymerase from Thermus aquaticus present, and so you basically will synthesize a new strand of DNA using these primers as starting points for the synthesis of the DNA. And then you just keep repeating these processes, okay? So you repeat it, this is cycle 2. Now you've got your new products, you've heated up the DNA, the new products are separating from the strands that they were originally hybridized to. You anneal your primers to the sequences, you go through another round of DNA synthesis. And then we'll go on to cycle 3 now. Cycle 3, you heat up the DNA, so it's all now been converted into single strands. You hybridize your primers or anneal your primers, and then you synthesize DNA again, okay? So I'm going through this fairly quickly, but you just want to make sure as you're looking through this, if you're not familiar with PCR, that you understand what's going on here. You can look at where the strands come from. I've got them color-coded so you can see what's taking place. Okay, so let's go on to cycle 4. We're going to heat the DNA up again. We're going to anneal the primers. We're going to synthesize DNA. And this is what we have after 4 cycles, okay? So if you look closely at this, you'll see after 4 cycles, half the products are DNA fragments of a very specific size. And in fact, they're exactly the size of the DNA that lies between the 2 primers. And it's these products that are going to be growing exponentially throughout the reaction. All other products that are not of this size, they're just growing linearly. So they're going to grow at a very slow rate, while these ones that are the specific size are going to grow exponentially. So after n cycles, if you do the math, there will be 2 to the n minus 2n products of this size, the size that I'm highlighting here. And because 2 to the n minus 2n is basically about 2n when n is large, and typically people do 30 to 40 cycles of PCR. So basically, if you have a billion molecules and you subtract 150, it's still a billion molecules, right? So 2 to the n minus 2n is basically 2n when the n is like 30 to 40, something like that. So, and again, that's the number of PCR products of heterogeneous length. That ends up being insignificant over time. You don't even see them because they don't ever rise to high enough abundance to be detected with any method that people use to detect the products of PCR following. Okay, so this is an exponential growth phenomenon. So after 30 cycles, like I said, you end up with many, many copies of the DNA molecule, even if you just start with 1 at the beginning. So following the PCR, typically what people do is they actually separate they take the PCR products and they run them on an agarose gel, which separates DNA fragments by size. So we'll show that on the next slide. So we're going to separate by size, and the question is, which of these bands, A, B, C, and D, which I've highlighted to the right of this agarose gel, is the largest? And basically the way this works, if you haven't done agarose gel electrophoresis, is basically you load samples in these wells at one end of the agarose gel, and then because DNA is negatively charged, you place this gel in an electric field, and the DNA will migrate towards the positive pole, okay? So which of these bands, and I'm showing you the products of PCR from 2 different individuals, so the question I want you to address is which of these bands is the largest? So if you have done this before, basically what you know is that in fact the largest band is going to be band A, because the agarose gel sort of separates these DNA fragments by size. The smaller DNA fragments can snake their way through the gel very quickly. the larger fragments have a much harder time snaking their way through the gel, so they move more slowly in the electric field. So band A would be actually the largest of the bands, whereas band D would be the smallest of the DNA products of PCR. So one thing that you might want to think about is, why do you see two bands in each individual? And it's because these variable number tandem repeats are highly polymorphic. It's not unusual for them to be different in size amongst the two different homologous chromosomes. And since you have two copies of each of these sequences, typically you're going to have two different alleles, although you can see cases where an individual is homozygous for the same allele length. Okay? Okay, so let's talk a little bit more about the primers that are used for PCR, and let's show what's really going on at the nucleotide level when you do a PCR reaction. So the primers must point inward to the region to be amplified, okay, and that means pointing inwards in a 5' to 3' direction, which is the direction that DNA synthesis occurs in. So let's say we have a double-stranded DNA molecule, and again, this is something I think you learned in the first half of the course. The two strands are oriented in a 5' to 3' fashion in an opposite way. The top strand goes in one direction and the bottom strand goes in the opposite direction. So let's say this is the region that you want to amplify. So what you would do then is you would basically synthesize your primers to amplify that region. So the 5', the primer that's on the left, we should go run 5' to 3' as it's shown, and the primer on the right should go 5' to 3' pointing in the opposite direction. So the one on the top that's pointing in the 5' to 3' direction is typically called the forward primer. The one on the bottom is typically called the reverse primer. Now, if I flip this DNA molecule over, then these names would change. So it's just the names are according to the direction you have that you're looking at the DNA, basically. Okay. So let's put a DNA sequence on here so we can actually look more closely at what's taking place. So again, as I told you, you're going to have primers are going to flank the regions that you want to amplify. So let's say the parts that I've highlighted here are actually the primer sequences you're going to want to use. You denature the DNA, so you have the two different single strands, and then you hybridize your primers to this region. So, and the way the primers are going to hybridize, again, they're going to hybridize in a 5' to 3' direction, because you want DNA synthesis goes in a 5' to 3' direction. And then you basically you have the Thermus aquaticus polymerase present and all 4 DNTPs, and you synthesize your new strand of DNA. Okay, so that's how the process works. And now I've got a practice question for you. So let's say we have the DNA sequence from exon 1 of the Huntington gene, and what 6 nucleotides from the blue boxes could be used as primers to amplify the region of the genome shown above? So you can stop the video at this point and think this through, but I want you to tell me what the left or forward primer is going to be and what the right or reverse primer is going to be, what the sequences will be. And again, I'm oversimplifying this. Typically the primers would be 17 to 20 nucleotides, but I'm making these very short, just for illustrative purposes. Okay, so hopefully when you looked at this, you sort of said the left or forward primer is going to have this sequence that I'm showing you here, and the right primer is going to have the sequence that's shown in the bottom strand, okay? And now the next question is, what size will the DNA fragment be after PCR amplification using the 2 primers above? So you can take a moment to count up the number of nucleotides to see if you've got this right. So I think in past times when I've asked this question in class, it seems like about half the people get the answer correct and half get it wrong. So I think the people that get it wrong tend to count the number of nucleotides that are between the primer sequences, but the products actually include the primer sequences in them, so you have to include those in your number. So if you've done this correctly, you should get 39 base pairs. Okay, so that's all I have to say about the polymerase chain reaction. Now we'll switch and talk a little bit about hybridization, the other method that's typically used to map polymorphic sites in the genome. Okay, so as I mentioned, the polymerase chain reaction is typically used to map VNTR loci, but the main method that's used to map polymorphic sites, primarily SNP alleles, is hybridization with allele-specific oligonucleotides. So let's talk about the principle a little bit here. So let's say you have a double-stranded DNA sequence like this, where one site in that double-stranded DNA sequence has a mismatch. It's designated by that little portion that's kind of stuck away from the that's not really you don't see a line connecting it to the bottom strand. So is this going to stably hybridize to the sequence that it's base paired with? And if the DNA sequence is extremely long, on the order of hundreds of base pairs, the answer is yes. But with short oligonucleotides, under conditions of appropriate conditions of salt and temperature, this would not be stable. Okay, this would not work. If there's a single mismatch in the sequence, it would not remain stably bound to its complementary sequence. It has to be a perfect complementarity for it to hybridize properly to its complementary sequence. And that's the basis of the allele-specific oligonucleotide hybridization. So the idea is you hybridize the small oligos that are 17 to 28 bases in length, and you basically only see hybridization if the target and probe match perfectly. Okay, so for example, let's talk about this sequence here. Let's say that there's a site in this DNA sequence designated by the question mark that varies in the population. You might not even know what sequence is present there, okay? And you can determine what that sequence is by asking if you generate 4 different probes. So let's say we denature the DNA, we generate 4 different probes that are complementary to the sequences that we do know what the sequences are, but they vary in that one base that's present in the very middle of the oligonucleotide. So you have all 4 different possibilities. Some oligonucleotides have the T, some have the A, some have the C, some have the G at that site. And then basically you just ask which of these 4 can stably hybridize to your target DNA. And based on which one hybridizes to the target DNA, that tells you what the sequence is at that site that you're trying to interrogate. Okay? And we can call these different oligos the G oligo, the C oligo, the A oligo, and the T oligo. That's sort of the terminology I'll be using in a lot of the slides that follow up. And again, the only difference between these is that they vary at that site that we're trying to interrogate. The rest of the sequences of all these oligonucleotides is identical. And only the one with perfect complementarity is going to hybridize to the target sequence. So this is one way you can actually determine what the sequence is, but typically you don't just take your DNA and mix with it a bunch of different oligonucleotides and ask which one hybridizes to it. The more convenient way to do this is to take the oligonucleotides that vary at this middle position and stick them onto a slide in some fashion so that the different oligonucleotides can be distinguished from one another. So that's actually illustrated on the next slide. So it typically involves taking a DNA sample and hybridizing that DNA sample to a slide or a membrane that contains all 4 different oligonucleotides in different spots on that slide or that membrane. So in the membrane that I'm showing you here, or the slide that I'm showing you here, you can see there's a portion of the slide that has just the T oligos, a portion of the slide has the G oligos, a portion of the slide that has the C oligos, and the portion that has the A oligos. And these oligos are actually covalently attached to the slide, so they're stably situated at that position on the slide. And the way this method typically works is you'll take in a DNA sample from an individual or from whatever organism you're working with, and then you fragment the DNA into smaller pieces, and then you take these fragments and you label them with a fluorescent marker of some sort. You heat them up to convert them into single-stranded form, and then you hybridize them to your oligonucleotide array, and you basically ask which of these spots do these fluorescent bands or fluorescently labeled DNA, which part of this membrane do they hybridize to, and that would tell you what the sequence is at the site that you're trying to interrogate. Okay, so let's look at an example of this use of oligonucleotide hybridization. So let's say you've got a mother and a father and a baby, and you want to genotype them at one specific site in the genome that you know is polymorphic in the population. So what you do is you collect DNA samples from these individuals. You just collect the cells, you label the DNA, and you hybridize them to oligos, just as I showed on the previous slide. And let's say you do that, and you see the following results. So this is the green is supposed to represent hybridization at these sites, and you can see on the left here that there's the A oligos at the very top, the T is at the very bottom. Okay, so based on this hybridization pattern, we can begin to infer what the genotype of these 3 individuals are. So if you look at the father, you only see hybridization of the A oligo. So that tells you that the father must be an AA homozygote. If you look at the mother, you see that you only see hybridization at the G to the G oligo. So she must be a GG homozygote. And if you look at the little baby, the baby has is an AG heterozygote, okay? And that's exactly what you'd expect because these oligos, these SNP inheritance is Mendelian. So the father only has an A allele to transmit to his offspring. The mother only has a G to transmit to her offspring. So the child has to be an AG heterozygote, okay? One of the nice things about using molecular markers in your experiment is that they're codominant. This may be a term that you were exposed to in the first half of the class, but what that means is the phenotypes of both alleles are seen. So you can see both alleles when you use molecular markers. And the nice thing about this oligonucleotide specific hybridization method is you can do this in a high throughput fashion. You can actually interrogate many different polymorphic sites at the same time. Okay, so we'll talk about that in just a moment, but first I'm going to ask you a question. And here's the setup. This is a little bit out of date. You guys probably have not seen this show, The Jersey Shore, but it's okay. I'm just going to sort of go through the question anyway, and you'll understand the logic. So you make oligonucleotides that specifically hybridize to a polymorphic SNP on an autosome. And so here's the setup. And this is the results of the hybridization, but here's the setup to the question. After the show was canceled, Snooki, individual 1-1, and Polly D, individual 1-2, developed a relationship and had 4 children, which is generation 2. They had 4 children together. Polly D has become suspicious of Snooki. She is courted by and courts a lot of males. Using only the SNP genotype data, has Snooki been faithful to Polly D? So I'm going to let you look at this, and you can tell me whether the pattern of hybridization that you see amongst the 4 children is consistent with the genotypes of individual 1-1 and 1-2. And when you're ready to answer your question, you can restart the video again. Okay, so hopefully after you looked at this, you concluded that in fact Snooki has not been faithful to Polly D. And the one individual in here that tells you that that's the case is individual 2-2. So basically in each of these individuals, the offspring of 1-1 and 1-2, they have to have inherited one allele from 1-1 and one allele from 1-2. But if you look at 2-2, 1-2 did not transmit anything to individual 2-2. Okay? So that's not consistent with that child being the offspring of 1-2. Okay? Now, if I had not told you that this SNP was on an autosome, you would not have been able to rule out this individual as the offspring of 1-1 and 1-2, because as you know, if it's an X chromosome linked SNP, males only inherit their X chromosome from their mother. So this could have been a male, and it could have inherited that one allele from the mother. So that's something you want to pay attention to when you're looking at problems, for example. Okay. Okay, so again, as I mentioned several times, the big advantage of hybridization for identifying SNPs, or genotyping SNPs, I should say, is that it can be done in very high throughput fashion. So again, the way you do this is you just basically label your genomic DNA, you hybridize to oligos for a set of SNPs. So let's just imagine we were looking at 10 different spots along the length of this particular chromosome that we know each of these 10 spots is polymorphic in the population. So in other words, you see variation at these 10 spots. Okay? So what you can do is you can create a SNP chip where you've got oligonucleotides that's specific to each of these 10 spots, but each of these oligos only differ from one another in the fact that in the base that you know is polymorphic at these 10 different spots. And then you take a DNA sample from an individual and basically you hybridize that DNA sample to this chip, and you're basically going to get results that'll tell you what the genotype is at all 10 of these spots at the same time. So let's say we do this. We'll walk through the logic together. Let's say we do this and we get the following hybridization pattern. So red is supposed to represent hybridization. So now you can just read off this chip. You can read the genotype at each of these sites. So if this is an autosome, for example, then you know that there have to be two alleles for each of these sites, so you can just read off the genotype. So at locus number 1, for example, the genotype is at number 1, it's a GG homozygote because you only see one hybridization, one spot that's hybridizing. In number 2, it's got to be an AA homozygote. At number 3, it's an AG heterozygote at that site, and so on and so forth. So again, the advantage of this method for genotyping SNPs is that you can take a DNA sample, label it fluorescently somehow, and then you can hybridize it to a whole bunch of different oligonucleotides that are located that correspond to sequences all through the genome, and you can get genotype information at many, many sites all through the genome all at one time. So as it turns out, I'm showing you an example of just genotyping 10 different locations in the genome at one time, but there's commercially available SNP chips that can detect up to 5 million SNPs. And in fact, this slide's a little bit out of date. You can actually detect way more than 5 million in a current SNP chip at this point in time. So you can sample many, many polymorphic sites all throughout the genome all at one time. So these SNP chips are actually quite small. Here's an example of ones, the ones that were contemporary maybe 3 or 4 or 5 years ago, something like that. They're quite small, and basically you're sampling 5 million on these particular SNP chips. This is what the hybridization pattern looks like if you zoom in on the part where the chip where the hybridization is taking place. Okay, so now I have a practice question for you. You're going to apply some of the things I just taught you to a problem here. Okay, so I'll read the question, and at multiple points you can stop the lecture video and you can see if you can get the right answer. So a family, mom, dad, and 3 kids, were genotyped at 6 different polymorphic loci on the X chromosome. So bear in mind it's the X chromosome, using allele-specific oligonucleotide hybridization. Now at each locus, the 4 bases being tested are A, C, G, and T, reading from top to bottom, and hybridization is indicated by shading and the base that actually was detected. No hybridization is indicated by blank boxes. The results for the mom and the 3 kids are shown below. Okay. So I have 2 questions I'm going to ask. There's a third question I'm going to throw in here also. So the first question is, one of the 3 kids is a boy. Which one is it, and how do you know? I'll just ask the second one right now too, and you can stop the video and answer both of these questions if you'd like. One of the kids is adopted. Which one, and how can you tell? Okay, so if you want to answer these on your own, you want a chance to think about it, stop the video now, and you can start it up when you're ready to see the answer. All right, so the answer to A is the one that's a boy is kid number 2, and the reason you know that is because the boys only have one X chromosome, and you only see one hybridization signal from each of these loci for kid number 2. With the other 2 kids, you see multiple hybridization loci for some of the loci. So kid number 2 must be a boy. And then what about which of the kids is adopted? Okay, and how can you tell? So once again, the answer is kid number 2. And the reason you can tell is if you look at, for example, just look at locus 1. So at locus 1, kid number 2 has to inherit at least one of those alleles from mom. And what you can see is kid number 2 has inherited a T allele at locus 1, but mom doesn't have a T allele at that locus. So that cannot be the mom cannot be the mom of kid number 2. And there's other inconsistencies as well in this pattern of hybridization. All right, so now a third question we're going to ask, so it doesn't match mom's genotype. A third question we'll ask is, you can fill in the appropriate boxes to indicate the dad's genotype. And once again, you can just stop the video and you can do this on your own. I've crossed out kid number 2 to make clear that you don't want to use kid number 2 to figure out what dad's genotype is. Okay? All right, so you can stop the video if you want to do this on your own, and then you can restart the video when you're ready to sort of see the answer. All right, so if you've done this correctly, what you should say is, at locus number 1, dad's genotype must be a G. And remember, dad only has one X chromosome, right? So there's only going to be one hybridization, one spot that hybridizes for each of these loci. So dad must have a G there, because if you look at, for example, kid number 1, you'll see that kid has hybridization signals for A and G, but mom can only transmit the A hybridization signal. So they must have inherited the G allele from dad. And just using this logic, you can say that locus 2 must be a G in dad, and locus 3, 4, 5, and 6 are as follows. So you just want to make sure you understand the logic that I use for answering this question, and just to make sure that you understand how to do this on your own as well. Okay? All right, so now Okay, so here's another problem, and this is a problem from problem set number 7. It's actually problem 11 from problem set 7. So in the past, I found that students have a very difficult time answering this question because they're not really quite sure where to get started with it. So I'm going to answer the first part of this question, and I'm going to try and set up the logic that you need to use to answer the remaining parts of this question. So just to read the question, the pedigree shows inheritance of Wilson's disease, an autosomal recessive trait, and of PS1 and PS2, which are two single nucleotide polymorphisms in the genome. And what I'm showing here is the SNP genotypes were detected using hybridization to allele-specific oligonucleotide microarrays, and the shading indicates hybridization, and the results are shown below each of the individuals in the pedigree. So in the boxes next to 2-1 and 2-2, what I ask you to do in the problem is to fill in their parental type genotypes for the three loci. Okay, and what I asked is to see the genotype given for 1-1 as an example. But what I'm going to do is I'm going to construct the genotype for 1-1, and then we'll talk, and I'll answer the question for individual 2-1 here, and then I'm going to let you do the rest of the problem on your own. Okay, so let's look at what the genotype is for 1-1 to kind of sort this out first. Okay, so for individual 1-1, this person does not have the disease, so they must have the dominant non-disease causing allele, the disease gene. So that's one part of that person's genotype. Whether they're homozygous for the non-disease causing allele or whether they're heterozygous at that site, we don't have enough information to answer that question. What we also see is with polymorphic site 1, we see that at that site the person's a heterozygote. They have an A allele and a C allele, and at polymorphic site 2, that individual is a GG homozygote. Okay, so that's the genotype information for this individual. So we can write that down. Again, the person carries at least one dominant allele, the non-disease causing allele. We don't know if that person's a homozygote or a heterozygote. The person's an AC heterozygote at polymorphic site 1, and that person's a GG homozygote at polymorphic site 2. So just a couple of points on here. I'm not yet connecting this line because we don't yet know if the polymorphic sites are linked to the disease causing allele. So I'm not going to connect the lines, but I am going to and we don't know anything about the phase at this point in individual 1-1. So all we know is the genotype at this point. That's all I'm writing down here. Okay. All right. So now, based on the information here, you can use the same logic in individual 1-2. I'll let you do that on your own to figure out what the genotype of that individual is. And now we can use the information that's given here for 1-1 and the information for the genotype provided for individual 1-2 that you can work out on your own to determine what the parental types would be in individual 2-1. Okay, so let's think this through. So individual 2-1 does not have the disease, so they must have inherited the dominant non-disease allele from individual 1-1. That person is an AC heterozygote at polymorphic site 1, but only individual 1-1 has a C at polymorphic site 1, so they must have inherited that C allele at polymorphic site 1 from individual 1-1. And then at polymorphic site 2, this individual is a CG heterozygote, but individual 1-1 only has a G to transmit, so that person must have inherited the G allele at polymorphic site 2 from individual 1-1. So one of the parental types is big D, C, G. Okay. Again, I'm not connecting these by line because I don't yet know if they're linked, but that would be one of the parental types. That's what's inherited from individual 1-1. And then using the same logic, I'm not going to go through it here. You can determine what the parental type would be that the individual 2-1 inherited from individual 1-2, his father, or her father, I should say. And that's what you should get if you work through that logic. Okay, so in the next part of this, basically what you need to do, I'm just explaining what you need to do. I'm not going to do any more of this problem. What you need to do is you need to consider both markers independently because, as I said, these are just these two polymorphic sites that we're considering here. They're just sites of variation in the genome, and they may be, one may be linked to the disease trait, both may be linked to the disease trait, maybe neither of them linked to the disease trait. I just haven't given you any information. So you have to consider them independently from one another. One may be linked, one may not, maybe neither of them are linked, but what I want you to do is consider them independently, okay? So assuming PS1 is linked to the disease, is the gamete received in generation 3 a parental or non-parental? Then do the same with polymorphic site 2. That's basically what I'm asking you to do in this problem, okay? Hopefully that's enough information for you to answer the question. I will provide answers in the end, of course. So if you don't get the right answer, you can always check your answers against the answer key. And of course, you can always post questions to the message board or email if you have questions as well. Okay? All right, just to summarize what I've told you up to now, molecular markers are just variant forms of DNA sequence that can be used to map gene locations. Polymorphisms include things like single nucleotide polymorphisms and length polymorphisms. And alleles of polymorphic sites show Mendelian inheritance, and they're codominant. Again, codominant meaning that you can actually detect both alleles. And alleles of polymorphic sites can be detected using a variety of different methods. The methods we talked about today so far have been DNA hybridization, PCR, and typically PCR is followed by gel electrophoresis to look at the length polymorphisms. Okay, so now we're going to talk a little bit about how you use these molecular markers in mapping experiments. And I'm going to use, again, an example from fruit flies. All right, so again, DNA polymorphisms are just simply genomic landmarks. They're like mile markers located throughout the genome. We don't know where the gene for our trait of interest lies, but if we can show that our trait is linked to a DNA polymorphism that we know is located somewhere in the genome, then that tells us roughly where the gene of interest lies in the genome. And as I'll talk about in the next lecture, once you know where the gene is located, that can help you ultimately identify the gene that you're interested in. Okay, so again, I'm going to use an example of gene mapping using a DNA polymorphism in fruit flies. So let's say we sequence a small part of chromosome number 3 from fruit flies, and you find that the two homologs at chromosome number 3 have a single nucleotide polymorphism. In other words, a site where the one nucleotide is different on the two homologs, and one of them has a TA base pair and the other one has a CG base pair. So I'm just going to come up with names to designate these different alleles. One of the alleles I'm going to call DM3TA, and one is going to be DM3CG. The DM stands for Drosophila melanogaster. The 3 is because it's on chromosome 3, okay? And the TA and CG designations just distinguish the two alleles. Okay, so these are alleles at this locus, and they may not even be coding for anything. They just might be in intervening sequences and introns. They could be anywhere in the genome, basically, okay? The important point is that it's a polymorphic site. It's a site where you see differences in different individuals in the population or between different homologous chromosomes. Okay, so the way we determine the genotype applies to this molecular marker is we just do a DNA test for the DM3 locus, and suppose you detect DM3TA and DM3CG. What would you conclude from that? And what you should conclude is that one homolog had DM3TA allele and the other homolog had the DM3CG allele. So that would tell you that this fly is heterozygous for this DNA marker. So one of the homologs has the TA allele and one has the CG allele. So how do we start looking for linkage? How do we test for linkage in general? What kind of a cross do we set up? So in general, we take a dihybrid for both of the traits we're looking at, and we cross it to a homozygous recessive. We do a test cross, right? So the logic we use using molecular markers is basically identical. What we do is we take a heterozygote, a fly that's heterozygous for the marker allele and heterozygous for the trait of interest, and we cross it to a homozygous recessive. Now, recessive doesn't have any meaning with respect to these molecular markers, but the important point is it's homozygous. That's the important point. And if it's the trait of interest that you're looking at, ideally you're going to work with the homozygous recessive of the trait of interest as well. So in our example, we'll ask whether the DM3 molecular marker that we've been using in our example is linked to the purple gene in Drosophila. So again, this is an example using Drosophila. We talked about the purple gene before, so you should be familiar with the phenotype of the purple gene and the genetics of purple. But this method, again, could be used and applied to a variety of different model systems, including humans. Okay, so let's say we have a true breeding red-eyed fly, and we do a DNA test, and we detect only the DM3TA allele. And we have a true breeding purple-eyed fly, and we do a DNA test, and we find that it only has the DM3CG allele. So whenever I say true breeding, that means you're homozygous for all loci, okay? And so we can actually write out the genotypes for the true breeding red-eyed fly, and the genotype of that fly would be homozygous for the dominant allele purple, because the recessive allele is what confers purple eyes, and it's homozygous for the DM3TA allele. And we also know what the genotype is for the true breeding purple-eyed flies. That genotype would be homozygous for the recessive allele purple and homozygous for the CG DM3 allele. Okay, so we cross these flies together and we generate the following offspring. And again, I'm not connecting the line here because we don't yet know if the DM3 allele is linked to the purple locus. But I am trying to illustrate the parental type here, right? Because the purple plus in DM3TA have to come from one of the parents. If they are linked, then the parental type is purple. One parental type is purple plus DM3TA, and the other parental type would be the recessive allele purple and the DM3CG allele. Okay, so this is our heterozygote. So now, when the heterozygote makes gametes, I basically already answered this question. What would you consider the parental types among these gametes? Okay, hopefully this is clear because we've been through the logic of this on preceding lectures. And this is what I just got through telling you. These would be the two parental types. Okay, so let's say we take this heterozygote and we do a test cross. And this is again the test cross that we would do. We take our heterozygote at both loci and we cross it to a homozygote for both of the loci. And with the trait of interest, it's the homozygous recessive. Okay, and then we score the progeny. And for each progeny fly, we'd ask what's the eye color and what allele is present at the DM3 locus. And let's say we get the following results. We'll go on to the next slide. So we get flies with the indicated phenotypes. So we'll just start with the very first entry in this table. Flies with red eyes, and they have the DM3TA allele and the DM3CG allele. So they're heterozygous for those alleles. So based on that, you should be able to say, and based on the cross that we did, you should be able to define the genotype here. So if you want to kind of fill in this entire table, you can stop the video now and fill it in. Or you can just wait until I get to the other parts of the question. It's up to you. So the genotype of that fly should be purple plus. It's heterozygous for the purple allele, right? Because it has red eyes, it had to inherit the dominant allele purple from the dihybrid fly. And because it has the TA and CG alleles of the DM3 locus, it must have inherited the TA allele from the fly shown at the left, and it had to inherit the CG allele from the fly shown at the right. Okay, and you can basically do all the rest of these on your own. The genotypes of the remaining flies are shown. You should be able to work all this out on your own. And then the next question we're going to ask is, what were the gametes that came from, for example, from this dihybrid fly that gave rise to these genotypes? And so once again, this is something you should be able to work out for yourself. So in this case, the dihybrid fly is the only fly that has the wild type allele, the purple gene, and it's also the only fly that has the DM3TA allele. So that must be one the gamete that was transmitted to give rise to this fly. And then you need to also ask yourself, is this a parental or a non-parental chromosome? And again, I've already defined what the parental types are, but this is something you should be able to work out on your own anyway. So this would be a parental chromosome. And using the same logic, you can fill out the rest of this table. Now, one of the things I didn't sort of say that I should have probably said right from the start, if you look at the number of progeny, you already have very strong evidence that the DM3 locus is linked to the purple locus. And the reason that's the case is because what you see is two progeny classes are way more abundant than two other progeny classes. And the two more abundant progeny classes, the number of flies you see for each of those is about the same. And for the two less abundant progeny classes, the number that you see for each of those is about the same. So that's exactly the pattern you expect for linkage. So right away, you can look at this data and say the DM3 locus is probably linked to the purple locus. You don't have to do a statistical test in this case. simply because the numbers are so large that it would pass any statistical test that you try to apply to this. Okay, so based on this information, we can now interpret the results. And again, based on the numbers alone, the eye color gene is linked to the DM3 locus. So in other words, where this DM3 locus is present in the genome, the purple gene must be somewhere nearby. And we can also calculate roughly how far away it is by calculating what the map distance is between the DM3 locus and the purple gene. And again, we've been through the logic of how you do this. You just count the number of recombinant progeny, divide that by the total number of progeny, multiply by 100, and that gives us the number of centimorgans away that the purple gene is from the DM3 locus. So if we do this for this example, what we see is that there's a total of 800 progeny from this cross, and of those 800 progeny, 78 plus 82 are the recombinants. And so when you do the math, it tells you that the purple gene must be about 20 centimorgans away from the DM3 locus. Okay, so that tells us that the DM3 locus and the purple gene are linked, and that the purple gene lies somewhere nearby to the DM3 locus. So when you do this in principle, the question that might arise in your mind is, which polymorphic markers do you use to test for linkage? And the issue here is you really don't know ahead of time, so you have to do a lot of trial and error. So for example, you look for linkage between the disease gene and the polymorphic site number 1. Then if you don't see linkage, then you repeat with another polymorphic site, say site number 2, and you just keep repeating this process. But the advantage of molecular markers is using molecular markers, especially SNP markers, is that these markers are scattered all over the genome. There's lots and lots of them scattered through the genome. So you have many markers to choose from. The other thing is they can be rapidly genotyped simultaneously in a high throughput fashion. As I told you, and this is a little bit out of date, there are chips that can be used to map or to genotype 5 million human SNPs on a single chip. So you can basically, in just one time, you can actually sample the entire genome to look for linked markers and find linked markers just in one single experiment. Okay, so to summarize what I've told you up to now, and I only have one more thing to say at the very end of this lecture, genes can be mapped relative to each other based on linkage. Genes can also be mapped relative to known DNA positions by using DNA markers or polymorphic sites along the chromosomes. And thus these DNA markers serve as landmarks to establish the physical locations of genes in the genome. So again, in the next lecture, I'm going to talk about how getting you into the basic neighborhood of where the gene that you're interested in resides, that helps you identify the gene that you're interested in, how you can clone the gene. Okay, so going back to one of the slide I showed you at the very beginning of the lecture, I mentioned that there are several challenges with linkage studies in humans. And I told you I'd only talk about how we deal with 2 of these. One of the ones, what we just got through going through, was we talked about how you use molecular markers to map genes in humans. The other issue I said that comes up is that many meioses aren't informative. So you can't really fix that problem, but I do want to define what I mean by an informative meiosis versus a non-informative meiosis. So a test for informative versus non-informative meiosis, it's really asking, can we tell if the gamete was recombinant? It doesn't matter whether it was a recombinant or it wasn't a recombinant. It's just really asking if we can tell if the gamete was recombinant, okay? So if we can tell, the meiosis is informative. So yes, the gamete was recombinant, or no, not recombinant. Both are informative. If we cannot tell if the gamete was recombinant, then it's non-informative. That's really what it's asking. All right, so now I have a question. Okay, so here's a question that tests the concept of informative versus uninformative meioses. So the pedigrees below show inheritance of autosomal dominant trait, big D equals the disease, and little d equals the normal recessive allele. And the numbers that are shown in curly brackets indicate alleles of a microsatellite repeat polymorphic locus. So the numbers could refer to the number of repeats in that locus as determined following PCR of the locus and running out the results on a gel. Okay, so the question is asking, is the meiosis in 2-1, is it informative or uninformative? And there's 2 different pedigrees, and I want you to answer for both of those. So I'll just give you a little bit of advice here. what you want to do is you want to write out what the genotype is for 2-1 phase included based on what that person inherited from the parents in generation 1. And then you want to also write out the genotype for individual 2-2, although you may or may not be able to say anything about the phase of individual 2-2 because we don't have any information about the inheritance, what that person inherited from that person's parents. And then based on that information, and then you can write out the genotype of individual 3-1 to say whether that person inherited a parental or a non-parental chromosome, or if you can't tell. Okay, so you can stop the lecture now if you want to work this out on your own, and you can restart it when we're ready to go through the answer together. So hopefully when you wrote out the genotypes, you came up with something like this, where basically I'm showing the genotypes above the individual in the pedigree. So the genotype for 2-1, that person inherited the dominant disease-causing allele from individual 1-1, and that person must have inherited the 12 allele of the VNTR from individual 1-1 because only 1-1 has the 12 allele. Individual 1-2 does not have the 12 allele. Okay, and then the non-disease-causing recessive allele, D, must have come from 1-2, and the 13 allele, the VNTR locus, must have come from individual 1-2. So what I'm showing you here basically is the phase of these alleles in individual 2-1. And the genotype of individual 2-2 is actually very simple here because this person does not have the disease. They're homozygous for the non-disease causing allele, and that person is a homozygote also for the VNTR that has 13 repeats in it. And now if we look at the results of this cross between in generation 2, and we look at the genotype of individual 3-1, what you can see is that person has inherited the disease. That person has also inherited the 12 allele that must have come from individual 2-1, and the other chromosome must have come from individual 2-2. So since this individual 3-1 has inherited the disease and allele 12 based on the parental type in 2-1, that must be a parental combination of alleles that 3-1 inherited. And so that's an informative meiosis. So when I say parental, that means the same thing as a non-recombinant. So now let's go through the logic in pedigree number 2. So if you follow the same logic, so that's an informative meiosis. So in pedigree number 2, if you use the same logic to work out what the genotype is, individual 2-1 inherited the disease, allele, and allele 9 of the VNTR from 1-1, and they inherited the non-disease allele and allele 13 of the VNTR from individual 1-2. And then the genotype of individual 2-1 is as shown above that genotype, homozygous for the non-disease allele, and that individual carries the 9 and 13 alleles of the polymorphic loci. Now, if we look at the genotype of individual 3-1 in this case, the situation is a little bit more complicated. There's two different possible ways this person could have inherited this combination of alleles. They could have inherited recessive disease, a non-disease causing allele, and the 9 allele from individual 2-1. And if that was the case, that would be a recombinant. The other possibility is this individual could have inherited the non-disease causing allele and allele 13 from 2-1, and that would be a non-recombinant or parental allele. So because we don't really know which of these explains the genotype of individual 3-1, this is an uninformative meiosis. You can't tell whether recombination has taken place or if it hasn't taken place. Okay? So I have more problems like this in the problem set, so I would just suggest you work them on your own, and hopefully you can, by working the problems, that'll reinforce this concept of informative versus uninformative meiosis. That's it for today's lecture.